So I think I'm mainly here to stimulate some conversations. Don't want to talk at you. I just want to get us all in the same framework, the same point of view. I normally have a very cluttered conflict of interest slide, but I've recently decided to declutter it, and there's the URL if you want to find out all the companies. This is about anticipating the unexpected, and I'm going to take us through exponential technologies. You're probably all familiar with one particular exponential technology, but I'm going to tell you some that you may not be that familiar with, and their application to affordable disease prevention. We don't talk that much. We don't talk enough about prevention and diagnosis. Aging reversal and somatic enhancements, not germline enhancements. And then just briefly on some environmental questions that I think are quite interesting and use some of the same tools. And this includes carbon sequestration, zoonotic diseases, and what I call space colonies on Earth, but basically this means total recycling. In a microcosm, you can focus more on what's wrong with recycling than you can in a macrocosm. And I want to just point out, if it's not evident from every single one of my slides, has a component that is computing, component that's ethics and safety. This is safety engineering. It's a lot of what we do. And I just use the car as an example where all of these things contribute to the safety that we have, standards, isolation, testing, redundant systems, surveillance, and licensing. Just use those same words in all the biotechnology I'm going to talk about in just a moment. And there really is a lot of computing here that's being, and all the technology, both biological and computing, is iconized for conversational reasons. Okay. So an example of a kind of an ethics safety issue is a growing number of biotechnologies that are going out there without proper testing. That is, by proper testing, I mean clinical trials, FDA approved, ideally. And these include, these are actually these are actually the exceptions that are going through clinical trials, but the category of nutritional supplements and the category of probiotics do not require clinical trials. And I'm trying to champion that if we start doing it anyway, we can start building up motivation for everybody to do it. So these are some that are doing it in Fit Biomics, Elysium, and Metformin. Metformin was already been through clinical trials for diabetes, but the thing that's new is it's being in clinical trials for aging reversal. Now this is the exponential that I'm talking about. These are factors of 10 on the y-axis, and you've got kind of a Moore's law scenario from when I started in this field in around 1973, which is off the left-hand side. And like, you know, since this isn't really a law of physics or anything, it's just a trend line, it can change. And fortunately, it didn't plateau, which it could have done. It took off in around 2004. It took off at a much steeper slope. So they were going at Moore's law sort of rate, which is quite remarkable, you know, doubling every year or two. And then it went up, and it may still be going up. It's hard to say whether it's plateauing. It's got another 4 orders of magnitude left in it. And it is, in this case, reading and writing DNA. So reading and writing DNA allows us to read and write all kinds of things, all kinds of materials, all kinds of data, all kinds of things ranging from organs to ecosystems. And as an example, for the writing, you've probably all heard of Crispr, and Editas is one of the companies that we started to get Crispr into the clinic. We've also had next generation sequencing, which is in the clinic for non-invasive prenatal testing. And here is the $3 billion genome that we started with, or that we aimed for years and got roughly in 2004, is now $300 price cost, and there's a business model for $0. Nebula Genomics uses homomorphic encryption and blockchain to ensure privacy. We can have very high levels of privacy. The real problem with privacy is that you're all dropping your DNA right at this moment. So if you don't want your DNA to be out there, then you should not leave your home. That would be my lesson number one. And why did those slope change so radically? Why were we going to a doubling time of every 2 years to a doubling time of every few months? And it was not filling a room full of robots or people, which would just spend money faster, but it was miniaturization, same lesson as in Silicon Valley. But in this case, we're not miniaturizing in 2 dimensions by carefully placing things on a grid, but in 3 dimensions and completely mixed up. So it's a little bit more like multiplexing than microfabrication. And multiplexing was first used by Edison in 1874 for 4 messages on a telegraph wire, and then it's been used in many optical systems since then. But this is molecular multiplexing. Molecular multiplexing means that in one droplet, sometimes droplets so small you can barely see them, you can have hundreds to trillions of reactions going on in that same droplet in such a way that they retain their identity, and you can go through many steps of the reactions with a trillion at once. So for the same effort as doing one, you get a trillion. And that's reflected in the cost structure. And here's some examples. You don't need to look through all the examples, but it's just to show that there's some data in the literature. Now, therapies. I showed you those cost curves, which are just remarkable, 10 million-fold change over a decade, very fast. That's for reading and writing DNA. And some of that is in the clinic, but it is not in the form of therapies. In fact, some of the therapies I've contributed to, the general field of gene therapy, or genetic editing in some cases, is going through the roof. It is now, that category is the most expensive drug in history. It's over $2 million a dose. Fortunately, you only have to take 1 dose, but nevertheless, and it is cost effective relative to how those diseases were treated. These are serious Mendelian diseases, and this is better. It was compared to $4 million a few years ago. And some of these diseases are up to $20 million. And it's not clear, that's over a lifetime. It's not clear their lifetimes are getting longer, so the costs are going up, not going down. So how do we do this? How do we bring that down the way we brought down the cost of diagnostics to close to $0, and the way we brought down vaccines like smallpox vaccine is essentially $0 because it's extinct. How do we do that again? And I'm going to say it's basically 2 categories. One is prevention via genetic counseling, and the other is looking for very common diseases. Because if you have fixed research and development costs, and the denominator is the number of people that benefit, you want to get that denominator to be as large as possible. And the largest, most common disease, well, you'll see in a moment. Start thinking about what you think is going to be the biggest denominator. That'll be your homework for the next few minutes. First, I'm going to talk about the first one, which is rare diseases, which are individually rare but collectively common. About 5% of people born in almost every country, whether you're inbred or not, whether you have a family history or not, about 5% of us will have children that are very severely affected. Some of them will be late onset diseases. They'll be hitting us about our age now. And here are 2 examples of these genetic diseases, very serious diseases. You can see what this baby looks like with TaySachs. And sickle cell, you've probably all heard of that. It's one of the first molecularly characterized diseases. But they've had completely different outcomes in the clinic. The way they've been treated is very different. In fact, I wouldn't even say the clinic, in the world. So what happened is because of the experience of Rabbi Eckstein in 1983, where he had 4 of his first 5 children had this TaySachs disease. It should have been 1/4 at random, but it's random. And he said, we got to do something about this. And he formed an organization where you can get very simple genetic counseling, where everybody has a number. It's semi-anonymous. If you're engaged, you send your numbers in, and you find out whether you're compatible. That's better than having a TaySachs baby in some people's view, but it is still very bad news that the love of your life is incompatible and very often ends in non-marriage. An alternative would be and some of them are pregnant, and so that's termination is one of the options. Adoption, there are many options, having the child. But if you go even earlier, if you go to dating, then they only meet positives. They don't even have to know they're a carrier. They don't have to have their genome in their possession or anybody else's possession. It can be completely encrypted. And that's one of the things we're looking at. And the cost of this worldwide is somewhere around $1 trillion. It hasn't been that accurately estimated because there's a number of costs, like the opportunity costs for the caregiver, typically a family member, and the child. There's a huge opportunity, but the cost of preventing it is incredibly low, and that's where we want to go. So, different subject, hopefully equally provocative, and this is transplants. So the first transplant was actually done at my institution, or Harvard Brigham Hospital is affiliated closely with Harvard. And in my birth year, they did the first transplant, and it set the tone for all transplants since then until very recently, which is you have to have a match. It doesn't have to be an identical twin, but it has to be a very close match, and it's quite rare. It's called histocompatibility. That changed in about 2013 with CAR T cells. And I'm not going to tell you much about CAR T cells. That's not the point of this slide. It's not that these are amazing new tools we have to attack cancer cells using a modified immune system. It's completely designed, completely unnatural, but it is remarkable at harnessing the powerful immune system. The point of this is the second point, which is that this is universal. That is to say, you can take T cells from any one of us and give it to anyone else. And that wasn't ever possible before because you couldn't alter the donor or the recipient to be compatible. They had to already be born compatible. But now you can alter them, and you can alter them because of editing. And editing is, I mentioned CRISPR, but there are other ways of editing that we and others have been involved in. Let's just call it editing, that you can not only add genes, like adding this chimeric antigen receptor, which is the key to fighting cancer, but you can subtract genes. And that's what these little minus signs mean, is we can subtract, in fact, several of them at once. And those, by subtracting them, we make them more compatible so they don't cause the host versus the graft or the graft versus the host. They're immunologically compatible. They're even compatible with chemotherapy. So some of these things help them make it through chemotherapy. And this is this little girl was one of the first winners in this new technology in getting her B lymphoma cured. Now let's take this one step further. How can we? That's T cells. They're kind of like a soup. They're like cells in suspension. They're easier to engineer. When you get a hard, so-called solid organ, heart, kidneys, liver, lungs, eyes. These are, by the way, human and pig, left and right. They're very similar in size and in structure and in physiology. But the point is, these are very hard to engineer, except through the germline. And you may have noticed that germline engineering of human beings is not exactly a popular topic. But germline engineering of pigs is fine. And transplanting pig organs is fine. I mean, we already transplant pig valves routinely. Those are not organs. Those are a little more complicated T cells, but they're essentially dead by the time that they're used. And that means that as a child who has a heart valve defect grows, they have to keep getting replacements, and this is not good. While a natural organ will grow and exhibit feedback that regulates its size. And there are various ways of doing this: 3D printing, scaffolding, development, chimeras, and humanized pig genomes. And I'm just going to talk about the last one, humanized pig genomes, so you can see how far we've come in a very short period of time. So we invented CRISPR in 2013, and by 2015, we had already done 62 edits in one cell. Now, we had never done more than 2 edits in a cell before that. So this is a big step up, and more about that in a moment. But this is the laundry list of things. When this field was started 20 years ago, long before we were involved in it, we were only involved once we invented CRISPR. We were invited by almost all the pioneers of this field. 20 years and $1 billion commitment. And they got stuck at, basically, they wanted to do one gene, which is this first gene, and that wasn't enough. As it turned out, there were 43 different changes that had to be made, at least, and we've now made all of those. And there's some interesting things, like, pigs are not sensitive to a lot of the viruses that kill organs or that kill us. So that's one thing that we'll come back to. So we tackled all these, starting with these endogenous retroviruses, these porcine endogenous retroviruses. So you may not appreciate it, but every cell of every pig in the world makes these retroviruses from within. It's not a hygiene thing. It's not like they caught the virus. They actually, it's built into their genome. It's passed on from generation to generation. And they express these, and the viruses will infect human cells. And if it's an immune-compromised patient, this is like worst case scenario, because they're not ready to fight off these foreign viruses. And they might even provide an incubator where these viruses could evolve into a zoonotic disease, like think of swine flu or HIV or Ebola. Those are all people in too close contact, too intimate contact with animals that have viruses. So this is the most intimate contact you can possibly be, where you share a blood system. Anyway, so these are the team. As I go along, I'm going to shout out each of these. Luhan Yang is the one on the left. She's remarkable. She was a graduate student, postdoc in my lab, and now she's CEO of a company in China and CSO of a company in Cambridge that do the same thing and cooperate with each other, Kihan and Epigenetic. And these 2 science papers were largely her doing, along with Mark Guell and Dong Yu and others. Now so we took this big step from 2 to 62, and it was embarrassingly easy. It was just like, it worked first time. This usually doesn't happen in science. And it took 14 days, not of hard work, but of sitting the Petri plates in the incubator for 14 days, and then a little PCR. So when you give a mouse a cookie, they say they want more. So we said, oh, if 62 is so easy, let's go for more. And this is the team that did more. This is Corey Smith, and Oscar, Khalid, and Verena. And I'm not going to go through all the details here, but we decided to tackle an even bigger problem where more edits needed to be made in a single cell. So we're editing single cells and we're cloning them out. So we clone them to make a pig. I forgot to show you the picture that's representative of a whole herd of pigs in China, and a separate herd of pigs of a different strain in America that are ready for doing transplants. And in fact, we've begun transplants in the non-human primates at Massachusetts General Hospital, and they've survived for 9 months. So we're getting close to ready to start doing human transplants. Anyway, there are other systems where we want to make even more edits, and just briefly to tell you the cutting edge of this, we've now made not 62, but 13,000 edits in a cell in a particular category of genes. And this leads us to, we can become even more ambitious in terms of the things we want to change. We not only want to make organs available to solve the crisis, where you might have 1 million, 2 million people per year that could benefit from it, but we'd like these organs to be better than the ones you get from humans. They're going to be better partly because it's under controlled circumstances. You're not trying to time a histocompatible person in Arizona with a recipient in New York, and then you when it arrives, the surgeon has the patient ready to accept and says, oh, it's a little too gray, we can't do the surgery. These will be, that's one advantage right away, but there are other advantages, which we know how to make animals pathogen-resistant, cancer-resistant, senescent resistant, and I've already talked about the immunity, and even cryopreservation is possible, where we could have essentially a freezer full of organs ready to go. And these are examples of animals that do very well at low temperatures, even desiccation and low temperatures. And the bottom two are particularly impressive because you can store them at liquid helium temperatures. Some of the others don't, only get minus 14 centigrade. But those two, the Polypedillium and Tardigrada, are liquid helium and back, and they're fine as intact animals. So organs should be even easier. They are small, but they're not that small. Here's a project where I'm stating these without proof, but there's quite a bit of literature that we and others have contributed to for all of these, you might call enhanced organs. But you want your organs to be as good as possible. You don't want them to be falling apart after you get into the patient. Here's an example where we've taken an industrial microorganism and made it resistant to almost all viruses. And we hope very soon to make it resistant not only to every virus in such a way they can't even escape. We can make it resistant to 92% of viruses so far, 100% soon, by a method that's on good theoretical grounds, which is to change the genetic code of the host. You can change it in such a way the host is unaffected, but the virus is completely flummoxed in multiple places in every single gene of every single virus. And so we've done this successfully in one case so far, and we're expanding it to, we think we can do this for any organism that has a virus problem. But to be. Another type of enhancement that if it were in the germline would get everybody very worked up, and maybe even not in the germline, which is enhancement, in particular in behavioral and cognitive traits. But I'm going to make an argument that it may not be that far off, and it may not be that controversial, because we have an aging population. We have a population that is facing cognitive decline, and this cognitive decline is heart-wrenching. It's for the whole family. It's very expensive. It lasts, sometimes draws out for more than a decade. And you just hate to see the person who is so vibrant and full of brilliance and energy fall victim to this. And so here's some of the things where these are genes that are known to be involved. They're either involved in genetic counseling of human beings, like APP, presenilin 1 and 2, or these are things that aren't in gene therapies. These are not necessarily fixing a gene that's broken. These are things where you're doing something slightly synthetic that's overcompensating for a natural process like aging that in particular people goes awry. So you don't have to necessarily fix the Alzheimer allele, which these are examples of, but you can do something else. So these are in clinical trials for Alzheimer's. And then there's these three genes that individually and in various combinations have been shown to provide cognitive enhancement in animals on multiple tests. And here's an example of some of this behavioral data down below, where you can see the difference between the black circles and the white circles are with and without this gene therapy. Okay. We can also improve radiation resistance. With as few as 4 genes, we can change radiation resistance by 100,000-fold. That might be useful in space or something. So now we're going to switch over to almost all the experiments that involve human cells in my laboratory are using my cells. And that's because it's ethically better for students to work on me than for me to coerce the students into donating their axons and oligodendrocytes. Now, actually, I didn't donate my brain for this. This was from my arm. We took skin cells, turned them into stem cells. The stem cells can then turn into a variety of different cells, which you can control quite precisely now We have a recipe of generating recipes now where we can make any cell you want, and they will interact. So these are two different cell types, neurons and oligodendrocytes, where it wraps the axon. You see this beautiful wrapping, which is just like natural, but this is happening in the lab. And it's just like what happens in the white matter and the spinal column of your brain, where you want to send things really fast. Well, this is sending really fast axonal transmission. Not only do we have these two cell types, we have half a dozen more, including cells that form the vasculature. So before, we couldn't make organs much bigger than half a millimeter, and that's not what you would call an organ. But now they are vascularized, and they don't just get endothelial cells floating around in there. They form tubules that are exactly the right size to just let blood cells through. And when you implant these into animals, these are humans' organs, which you implant into animals to test them, because you don't want to test them in humans right away. They fill up with blood and they transmit blood normally. Even cooler, these organs, or organoids we call them, and trying to be modest because they aren't really perfect organs yet, but we can not only make normal-looking organs, but we can make abnormal organs, and we can make them really fast. This actually surprised me. We can make the organs quite quickly, but these abnormal ones, I thought, well, if it takes 70 years to get late-onset Alzheimer's, then who are we to think that we can induce an Alzheimer's model in human cells in a Petri plate or a more complicated organ system in less than that? But we can do it in 2 weeks, and we published this in this 2018 paper, and we've got some unpublished work on schizophrenia and bipolar, which is very similar. We can see the difference between the cells derived from the patient who is destined or has already gotten Alzheimer's versus age and gender matched controls. And I'll show you this is the only complicated data slide you're going to see, but you'll get the idea. I think you can see, even if I covered up the bottom of the slide, you could classify which ones are Alzheimer's on the right from the normal controls on the left. Now, there's a lot of differences between, we age and gender match them, but they're genetically very different. The difference between you and me is 3 million base pairs out of 6 billion. So there's a lot of differences. So we wanted to do that very carefully, and that's what we did on the right-hand side here. This is changing one base pair. Some of you may have even heard of this gene, the ApoE3 versus ApoE4. If you're ApoE4 homozygotes, you have about a 20 times higher risk of Alzheimer's than if you're ApoE2 or 3 homozygotes. And so we did that very carefully controlled, and you can see the same kind of result. Night and day, I don't even have to label the different samples. So that's interesting, not only because it's predictive power, but we now have, essentially we can engineer a human being without actually doing anything unethical. We're doing cells and then turning it into a piece of a brain that has these defects that we think are indicative of Alzheimer's. That means we can test out new therapies, we can test out new cause and effect rather than just correlation. These are all cause and effect experiments, because here we changed one base pair in the genome and saw the same thing we saw in patients. Okay, so I'm getting to I'm slowly building you up to thinking more and more about aging, ways that we might treat cognitive decline by cognitive enhancement, and that may result in off-label use for cognitive enhancement of people who are not in cognitive decline. Another type of enhancement is aging. We are not talking about longevity here necessarily, or at least that's not what we're going to go to the FDA about. Because if you go to the FDA and you say, aging is genetics. I know that because mice die at 2.5 years and bowhead whales live to 190 years. So I'm going to make humans live for 190 years. And the FDA is going to say, great, come back in 100 years with your clinical trial data, right? And so we're not talking about longevity. We're talking about aging reversal, which is much faster. It's been proven in several different animal models, different genes, different animals. We can get aging reversal. We can even get longevity. But there are 9 pathways. These are pretty well characterized. You could say half empty, half full. There's a lot more to do. but there's a lot we can do with what we know. Like we didn't need to know everything about smallpox before we started smallpox vaccines. In fact, we didn't know anything about viruses or immunology when we started smallpox vaccines. But we know a ton about these 9 different pathways, and you probably have to get all 9 of them right. So it may not be a simple drug or simple food or like caloric restriction. That's probably, you may have heard of this. You probably don't want to try it because you really need to be skin and bones to have this have the effect. But all of these things can be achieved not just by the way they've been achieved, but by going down into the cellular mechanism and skip the caloric restriction, eat whatever you want, but change what would have happened, what the body is signaling. You can short circuit that and engineer it. So anyway, that's what we've done. We've taken, Pedro de Magalis, who was a postdoc in my lab a decade ago, made a beautiful database, which is still the dominant database. And we took 305 human genes there and picked a subset of those which were particularly suitable for genetic gene therapies. And we took 45 of these, tested them in mice, and then found a set of 3 that would simultaneously solve 5 different diseases. So when you get 3 genes in one combination, so you can think of it as one treatment, it's a combination treatment, just like for cancer and infectious disease, covers them all at once. That's probably we're on the right track towards something that will cure all or most of the diseases of aging. So that we've now moved this on to dogs, because people care about their dogs. They try to clone them, and cloning them is kind of an insult to the dog because, this means I don't care about you, I care about your breed. You might as well go get a puppy of the same breed. But aging reversal gives you a few more years of the dog that you know. But anyway, the goal of this is also to go into humans as soon as possible. And Noah Davidson, I was just with him earlier today, who has a company right down the road here called Rejuvenate Bio. Okay, and the last topic is carbon sequestration. And this is a complicated, what I have to say is complicated, but it has some interesting, possibly slightly counterintuitive things. I'm going to tell you, we heard earlier today, and I hope I'm not being too controversial here, about how we're planting some trees, and that's great. And we're making biodegradable plastics, and that's great. But I'm going to tell you a scenario where we want to tear down the trees and we want to make non-biodegradable plastics. So the point is, it's not one size fits all. There are certain forests that need more trees, and there are certain forests where they have too many trees. So here's the deal. So a lot of effort is going into reducing our carbon footprint, and this is great as well, converting to electric cars maybe. But even if all the humans disappeared from the planet tomorrow, went to Mars or something, it would still be 10 gigatons a year. Sorry, that would remove 10 gigatons a year of carbon. But the amount that's at risk right now, and the amount we actually have to reverse, where we've already gone too far, and we would like to reverse about 100 gigatons, and the amount that's at risk without humans involved is 1,400 gigatons. That is what's melting in the Arctic. And the Arctic, some people just think of the Arctic as being a bunch of snow, but it's actually the thickest, richest carbon source in the whole world. In the Amazon, you might have a meter of topsoil. It's very thin. But in the Arctic, it's 500 meters, and my arms aren't big enough for that. It's almost a kilometer of topsoil in some places. And when it melts, it's turning into methane, which is 35 times worse than carbon dioxide, which is already pretty bad. So anyway, humans make 10, tundra makes 1,400, humans can leave, we still have, what's left is a problem. And so we need to do reversal, and we need to do reversal of both we need to pull the carbon out of the air, and we have to keep this carbon in the tundra, in the Arctic soil. And so 2 of the things that we're looking into is making virus-resistant algae. We need some kind of algae that will, either seaweed or cyanobacteria algae, that will fix the carbon in a way that we don't use up all the fertilizer, because they're starved for fertilizer, and the world is, in a certain sense, starved for phosphorus. And we want to segregate with this carbohydrates from the fertilizer. And there's various carbohydrates that are very hard to digest. For example, we have the USS Constitution is out in the Boston Harbor, and it's got the original wood in it, the wood that's in contact with the, so that's a non-biodegradable bioplastic. At least it can be made non-biodegradable. And so we're working on keystone species that were killed off not by evil corporate forces, but were killed off by our ancestors 15,000 years ago. Almost all the herbivores in Canada, Alaska, and Russia were killed off. And the consequences, the ecosystem changed from grass to trees. The trees are dark, skinny trees that bring in heat, or trap heat, and they also trap snow in the winter. So the heat raises the temperature in the summer, and in the winter, the snow is an insulating layer that keeps the -40 wind from getting down into the ground. And so if we were to switch back to grass, it would increase the albedo, the reflectance of the light. The grass is more reflectant, but it's also more photosynthetic. It's better at both of those. And in the wintertime, if you got rid of the trees, then the herbivores pack down the snow. This is well established by in an ecosystem engineering experiment. This is ongoing in Siberia with a group that we collaborate with, the Zimovs, their family enterprise. And it's very impressive what they've done in Siberia on a shoestring budget. And during our last visit to Siberia, not only did we spend time at the Pleistocene Park, which is this experimental place for changing the ecosystem by reintroducing herbivores and changing trees into grass, we managed to get a hold of 6 very high-quality mammoths. And this is Ariona Hisoli, one of my postdocs. And you might wonder why we were wearing matching clothes here. It's because there's a lot of meat flying around the room when we're doing this precise surgery. This is we use a big drill bit, basically. This is a cross section of one of its feet. No animals were harmed in this, by the way. These guys were 40,000 years old. They had a good life. But anyway, this is, I don't know how many people here have seen Fantasia. They have the little ballerina elephants dancing. Well, it turns out that elephants do actually stand on point. I don't know if Disney knew this, but this is their toe right here, and in the back is a bunch of fat. And so the cross-section of the foot is fascinating. I have comparative zoology background, among other things, and I was surprised by this. Anyway, we took samples of the bone marrow, the fat, and the muscle. And we came up with a new method that I won't go into for figuring out the genome. But we're not just reading the genome, we're also writing it. We're trying to make cold-resistant elephants that will reoccupy the space. Because of all the herbivores that have been brought back to Pleistocene Park in northern Siberia, none of them are capable of knocking down trees. Even though the trees are quite wimpy, they're about this big around after 50 years of growth. An elephant could just knock down a whole row of them in a few seconds. They love knocking down trees. I think it's their solution to getting the top branches rather than growing a long neck like a giraffe. Anyway, just finishing up here, get your questions queued up. There are more than 10 million square kilometers each for Arctic, pelagic, desert, and farm photosynthesis. Each of these has a different approach that we can get for fixing more carbon and keeping it fixed. You don't want it to recycle. You want it to stay in the soil, either frozen or somehow, or in the water where it doesn't degrade. This may be a little counterintuitive for those of you who haven't thought a lot about ocean ecology, but the richest biosphere is not near the equator or any of the warm waters, Florida, so forth. It's way up here in the Arctic. This is where all the rich life is. That's what this green and yellow and orange means. There's a lot of carbon being fixed up here. Unfortunately, it's all getting released again. So if we can even take a tiny fraction of that and keep it fixed, putting it into recalcitrant or non-biodegradable polymers, that would be great. Here's an experiment that was done where or actually this is a natural experiment that's done, but unnatural experiments have also been done of this sort, where you add iron to fertilize, and you can see it from space, these blooms that occur when there's an iron upwelling or intentional dumping a tanker full of iron into the ocean. And it's an incredible return on investment. Iron is limiting, and there's 1 million carbon atoms that are fixed for every atom of iron. So that's kind of cool. And this brings us to this business about space stations on Earth. How do we get sealed containers that illustrate recycling, perfect recycling? We've never done this for humans. We've done it for animals and plants. Here's one of the first ones in '72. You may have seen these little ecospheres. I encourage you to get one. These things last much longer than you would expect. Little shrimp inside here in an ecosystem with plants. But here's some of the human biospheres that have been attempted. Almost none of them were perfectly sealed. Almost none of them were considered a success. Here's my daughter in a submarine. Here is the International Space Station, which, believe it or not, is not a sealed environment. There's a lot of stuff going in and going out. They do not recycle very much. And they also don't, well, anyway, don't care that much about the calcium of these poor guys and gals. And Biosphere 2, this was a gigantic experiment, hundreds of millions of dollars, and it was so big that you couldn't really keep track of all the carbon. What we need are these smaller ecosystems that people, and more realistic as well, where you could imagine putting it on a spaceship. But we don't want to do these ecosystems for the first time on the moon or Mars, because the consequences of making a mistake are huge. While if you test them out, have hundreds, thousands of them on Earth, then if somebody gets sick or goes crazy or whatever, they can just walk out the door and go to the nearest hospital. And this is the very last slide. This is showing how some organisms grow faster than others, and so one of the things you want. So the one on the right-hand side was I'll do that over again. This is the previous winner for fastest growing organism was E. coli. You probably all heard of it. It's the mainstay of molecular biology. Here's the new winner, Vibrio. It's kind of scary how fast it grows. This is at exactly the same time-lapse speed. And then these are some of the photosynthetic organisms that also grow pretty quickly and have up to 60, 80% of their body is protein, for those of you who like protein. So I'm just going to say thank you to all these people along the way. Jim Markman is leading the project for transplantation at MGH, transplanting this in the primates and humans. And this is what we talked about. So hopefully I've stimulated some conversation now. Lots of good questions, I know, in this very technological audience. Thank you very much.